Welcome to this session in pharmacology. We're going to have a look at some discussions and some debates around the use of evidence-related research in the use of therapeutic medicines and devices. To enable us to do those discussions, we need to have a look at a few terms, and so in this session we're going to have a look at the way drugs are named. First of all, look at what we mean by a drug, and we'll see that we are talking about Western pharmacy, complementary medicines, CM, over-the-counter medications, OTC, and when we've seen these naming conventions, we'll then understand how that's used in publishing, how then that affects legal and ethical considerations with the registration of drugs. We'll look at other ways of classifying drugs, including this concept of scheduling drugs. And all of these aspects of publishing, reporting, registering, scheduling will involve studies on those drugs and their risk characterization. And once the risk has been characterized, we'll see that means looking at risks and benefits and weighing them up, that there will then be this risk management done by those legal registering authorities. Now, once we've looked at all of those important principles and seen how they parallel in both complementary medicine and in Western pharmacy, we'll look at two key principles that once again are foundational in both complementary medicine and Western pharmacy, these concepts of pharmacodynamics and pharmacokinetics. So first of all, what we mean by pharmacology, we're of course studying drugs, but the term drug we have seen includes prescription medications, whether they were prescribed by a general practitioner, a GP, or a complementary medicine practitioner. We'll also see that a lot of those over-the-counter medications, OTCs, are both founded in Western pharmacy and in complementary medicine. The study of drugs, of course, includes those that are legal, the ones that we've just been talking about, prescribed as therapies, but it also includes some legal substances that are prohibited in their access and those that are illegal. So we've seen that to say drug includes a whole range of natural and synthetic substances. These substances are sold or they are prohibited and regulated in their access because they have some action and effect on the living systems of the body. They're being sold as a therapy because they have a beneficial effect on the body, but it's important to realize that whether that substance is a Western pharmacy or a complementary medicine, it has the potential for some toxicity in the body, and all of these aspects are part of pharmacology. We'll see that pharmacology comes under the bigger banner then of toxicology studies. But another key concept is that pharmacology even has its foundations in pharmacognosy, as does herbal medicines. Pharmacognosy is looking at natural sources, plants, microbial toxins, animal tissues, minerals, other substances from nature, using them as therapeutic substances but trying to isolate and characterize the actual active ingredient in those mixtures, and perhaps to come up with formulations that are more beneficial as a therapy. And so that's the nature of Western pharmaceutical investigations and complementary medicine investigations, because they're all pharmacology. Once again, when it then comes to publishing information about those risks, the risk characterization, that information will be published under a particular name for that therapeutic substance. So this concept of drug naming is very important. We'll look some more at various generic and brand names of substances. This is the most important key principle here with naming, that in any of the literature, say MIMS Online, a particular publication available in Australia on Australian medicines, that it will use the approved Australian name, a generic name. It's often based on the chemical name, or it might be based on the action that that drug has, but it's an easier to remember name. It's a shorter name, certainly shorter than its chemical name. But oftentimes that generic name can be confused with a brand name. The brand name is just chosen by the company for that particular formulation that contains that generic substance. We'll also look in a little more detail at the use of classification systems for drugs, and so we'll use the major classification system classifying drugs by the body system on which they act. But there's other ways of looking at drugs, reporting on drugs, and therefore classifying those substances in the literature. This idea of scheduling we'll look at in some detail. It's another way of classifying substances. Safety and pregnancy is another classification system, as well as we've seen this system of classifying access to that medication, whether it's over-the-counter, not requiring a prescription, prescription medications, or those requiring a prescription from a complementary medicine practitioner, or over-the-counter medications sold through various access points that are actually complementary medicines. So to use an example of drug naming, we've chosen a Western pharmacy substance, amoxicillin. It is an example of an antibacterial antibiotic. When we go and study the classification antibiotic later, we'll see that there's some antibiotics that act against a whole range of microbes. Bacteria are just one type of microbe, so hence the further subclassification, an antibacterial antibiotic. Now in that name amoxicillin, that's its generic name. It's a simpler, easier to remember name, and it denotes that specific chemical substance that has the physiological effect. But also factored into its name is a little bit of information about the chemical class to which it belongs. So here's its chemical name, showing you how they are generally much, much longer than this easier generic name that is chosen. Amoxicillin is dextronegative alpha amino para hydroxy benzyl penicillin, but you can see in its chemical name that it's from the chemical class of penicillins. That will give you some information then about its molecular structure and therefore why it has its physiological actions in the body, but the Australian approved name is the name under which it will be classified in the literature, and its risk profile, its benefits, adverse effects and so on will be listed. When it comes to the proprietary or brand or trade names, we've given three here. We have Alphamox with a registered trademark, Amohexal with a registered trademark, Amoxil with a registered trademark. In the drugs acting on the body systems that we're going to go on and study, we'll use that registered trademark, or perhaps we'll put TM for a trademark. So this is a restricted name. It came from the individual company and they registered it under that name. There's a copyright for marketing purposes on that name, but whatever that proprietary name, they all refer to the same drug in that formulation, though the formulation itself may be different. So in this particular example, all three proprietary names contain amoxicillin in the required safe active dose. Now we're going to use generic names, as does all of the literature, in studying the drugs acting on the various body systems. There's other ways of classifying drugs, but important to realise that that's going to be the major system that is used in many of the textbooks, pharmacopoeia, electronic literature. So we've given an example here of a body system, the alimentary system. We'll talk about it as the gastrointestinal tract, and so here is a sub-classification of drugs that act on the gastrointestinal tract. Some of these names will be similar classification systems that are used in your complementary medicine modalities. But in MIMS Online, drugs acting in the GIT, we have hyperacidity, reflux and ulcers, drugs particularly working against those conditions. Then we'll have some that are classified by more their action. There's antispasmodics and motility agents. The term antispasmodic is one that might be a classification system also used in certain complementary medicine modalities, as would be laxative and antidiarrheals. This term digestive supplements and cholelitholytics, specific to, say, a pharmacopoeia or MIMS Online, but the term digestive supplements would also be used in modalities. Just incidentally studying the Latin, and similar to other studies in complementary medicine, you're taught to use Latin and Greek roots, prefixes and suffixes to analyse and work out what a name means. So these are cholelitholytics, drugs that will break up gallstones. And one last sub-classification that comes under the major classification, drugs acting on the alimentary system, we have topical anorectal medications. So it would appear in the literature, for example, MIMS Online, and would include a whole range of generic substances. So now let's have a look at some of these classification systems based on access to the medication. Here we have the over-the-counter medications, or OTCs. It's important for you to study these for several reasons, and here's a major one. Approximately 60% of all medications purchased are over-the-counter meds. So just for that volume of sales alone. But by that volume of sales, that implies that there will be a volume of potential adverse effects and drug interactions that these substances could be implicated in, and then thereby a whole volume of knowledge that complementary medicine practitioners will need to have about these substances. So this realization that your client will be buying these substances because they self-diagnosed and are self-treating. It may be a minor illness that has mild or self-limiting symptoms, and the adverse effects of this medication are known and published, and therefore you, the complementary medicine practitioner, can access that information under the generic name, or for certain electronic resources perhaps by a brand or trade name if that's all your client knew, and you can look up how truly safe and effective those substances are. Generally, these OTC meds are quite safe. There will be an incidence of adverse effects that are severe, but that is the case with all medications, even herbal medications. But that potential for harm will be low so long as other controls are put in place, proper packaging, concentration in that over-the-counter medication, limiting perhaps access or advice given when that medication is sold, controls around manufacturing and packaging, and clear information about dosage and safe dosage and so on. So we're not saying that they are absolutely safe, same as you wouldn't say that in your modality that herbal medicines are absolutely safe, that it's that you know the risk and it's low. Same with these OTCs. Now similarly, another parallel here with the manufacture of complementary medicines and these over-the-counter medications, sometimes the two areas, those two classifications overlap anyway, and both types of access to these medications come under certain quality controls. This particular one, good Manufacturing Practice Quality Assurance Systems, GMP. So this is where under these systems there's controls on the labelling, so that consumers, if they follow the directions on the label or can access the generic name on that label, will then not be so likely to experience adverse events or problems with dosage, overdosing on that medication and so on. Now, with some of these over-the-counter medications, we'll talk more about this aspect shortly. They should be sold knowing that they have a good efficacy for those minor symptoms for which the patient is largely, self-prescribing. However, this needs to be population data available on those over-the-counter meds to see that they do show that efficacy in the wider population. And we'll talk more about a related but distinctly different term, potency, shortly when we talk about safety and dosage of medications. Now, once again, over-the-counter medications we've said might overlap with a whole variety of other nutritional and herbal supplements and other complementary medicines. These are not distinct black and white categories, but OTCs largely will include not only nutritional supplements, but some of the classifications which we've seen are also part of Western pharmacy. So we have largely those OTCs that are sold are analgesics, antacids, laxatives, antidiarrheals, cough and cold preparations, and antihistamines. With nutritional supplements, we're saying 60% of all sales are for disorders where those drug classes are therapeutic. Now, you've seen another way of classifying drugs by the body system that they act on, and we've seen that antacids, laxatives and antidiarrheals could also be classified as drugs that affect the alimentary system or the GIT. Once again, some of the issues that surround access and sale of over-the-counter medications parallel some of the problems that might occur with complementary medicines. One of the major issues is this idea that clients self-diagnose problems and then access those medications to treat these minor symptoms. Any medication produces adverse effects, and even these over-the-counter medications will produce adverse effects and will interact with other drugs, so that this will affect how these substances are labelled, scheduled and so on. These over-the-counter drugs are marketed, so their marketing and the way that they are published and presented is also controlled by the Therapeutic Goods Administration, and we saw this was another parallel with complementary medicines, that their marketing and other aspects are also controlled by the TGA. With over-the-counter medications, we said that they should be sold such that they show good efficacy, but a lot of the times at the concentrations that they can legally be sold without a prescription, they aren't effective, they don't have this efficacy, they are merely potent, they still have a physiological action and work at the site in the body where they're supposed to. Now that then has certain ethical aspects of taking money from a person making therapeutic claims, but having to make sure that in your marketing and labelling you aren't making larger claims than you possibly can. So oftentimes they'll talk about substances that are OTC as being very, very potent, but they will avoid using the term efficacious or effective because they know that at that concentration they are not. Now, as we said, OTC as a classification could overlap with a whole variety of other classification systems, and some over-the-counter medications could be included in combination with some herbal medications or some dietary supplements and so on. So this idea of over-the-counter meds coming in as combination products means that there's a greater risk for adverse effects and drug interactions, because now your client is taking a mixture of substances. So once again, you need to know how to access literature and information about combination products so that you can see if there might be any individual ingredient. Is it in a dose that might be seriously interacting with a herbal medication that you may wish to prescribe? So as you can see, as in complementary medicine, these over-the-counter meds are important for complementary medicine practitioners to know about them, and they may be involved in that very important consumer education about ways to reduce the risk of adverse events such as overdose or adverse effects and so on with Western pharmacy medications. Now, as to the legal and ethical considerations, you will look at these in much more detail in your modality. Here we are looking at the parallels, that all of these substances sold as therapeutic under the regulation of the Therapeutic Goods Administration, and by the term goods we imply it doesn't just include drugs, it's medical devices that might also have a therapeutic effect. So thereby they come under Commonwealth laws, because the TGA is controlled by the Department of Health and Ageing, but there are also various state and territory laws that come into play with the registration of therapeutic goods. But through the TGA, therapeutic substances registered on this ARTG, Australian Register of Therapeutic Goods, and the parallels here, as we said, is the important aspect for you to realise that this register registers prescribed medications, listed medications, and by listed this is most of the over-the-counter medications that are low risk, non-prescription, but it also includes complementary medicines, those that might be non-prescription and those that are prescription. And just recently, in 2012, the Auditor General put out a report on ways into the future where the Therapeutic Goods Administration will work with complementary medicine practitioners in the ways that they will regulate complementary medicines into the future. So that TGA already involved in the registration of drugs that are complementary medicines to be used on humans. The TGA also registering other non-prescription meds and over-the-counter medications. TGA is also involved in controlling prohibited drugs. We'll talk more about these when we talk about scheduling, but this idea that one of the primary aims of the TGA is to control the supply of drugs that may be prone to abuse, thereby regulating availability of substances. It's not just based on their psychoactivity, but perhaps also any toxicity profiles that they may have, where there needs to be regulation around those substances. But also one of the other major aims of the TGA is to, in public health, ensure that there is ethical access to medications by the public, regardless of your socio-economic status. And so it includes certain schemes, such as this pharmaceutical benefits scheme, whereby registered therapeutic substances, some that maybe are orphan drugs, aren't yet adopted by an industrial company for marketing and manufacture, and a whole range of other substances can still be affordable and accessible by the people who need them. So whether it's a complementary medicine or a Western pharmacy, all substances have to be investigated and then that information reported to that TGA. So to do so, there will have to be some clinical trial data available on that substance, and there are certain regulations around the way that those clinical trials have to be conducted. If it's a complementary medicine that, because of its nature, doesn't lend itself to clinical trialling, then there is still this requirement for an exemption while it's being investigated, or if you are going to seek registration and then use of this substance, a notification scheme. But in general, most of these substances will go through clinical trialling, and this will be done by a sponsor company. This sponsor company must be an Australian entity. They have to be registered with the TGA as conducting that trial, and there are controls around the way that that sponsor then reports that information and how the data that is generated is managed and reported to the TGA. And it's important once again to realise that the TGA here is using international standards for these reporting protocols. Another important aspect, once again a parallel between Western pharmacy and complementary medicine, that if you are going to do any of these investigations or to seek registration and then have a notification scheme for any adverse events or risks and so on, that substance still has to perhaps go through some level of ethics committee approval. Certainly if it's a randomised controlled clinical trial, the ethics committee approval requirement and the data that will be required by that ethics committee is quite extensive. There might need to have been some preclinical trial data available on toxicity profiles of that substance and so on. Or if there's any other clinical trials that might be relevant, the RCCT, as we'll talk about shortly, being just the gold standard type trial, but other clinical trials might also be relevant for that complementary medicine or Western pharmacy medicine, that they will still need some level of ethics committee approval by that complementary medicine organisation or research organisation or that pharmaceutical organisation. Some of the preclinical data that might be used could include animal studies to avoid ethical issues with the overuse of animals for studies where these substances are to be used in humans. There's now certain technology-intensive tests, high-throughput screening tests that can be done on drugs to look at the probability of risks occurring and to study efficacy and so on. So this data, we're actually starting to talk about pharmacokinetic and pharmacodynamic data on complementary medicines and Western pharmacy. So we'll talk more about those shortly, but it's important to realise that this data is put to characterising the risks with that substance. It's a risk assessment procedure. So we're looking at risks versus benefits of that substance. Now that randomised controlled clinical trial isn't relevant to certain complementary medicine modalities, and we'll investigate that more in some of our debates and discussions. But the RCCT, when it is relevant, is needed for that complementary medicine or that Western pharmacy medicine to test its actual efficacy, to start to get an appreciation for the risks, that likely hazard, and likely, once again, being able to put a probability, a number on what percentage of a population may show that adverse effect and so on. Now, it's also important to realise, with Western pharmacy particularly, that even after it has then been registered and approved by the TGA, that there is still some clinical investigation being done on that substance. These are post-marketing studies. It might also be called pharmacosurveillance or pharmacovigilance. Also important then to realise that once a substance is approved by the TGA and marketed to the public, if any of that risk data shows that the risk versus benefit characterisation is that the risks far outweigh the benefits of that drug, then the TGA can withdraw the approval of that substance. And we'll actually, when looking at the drugs affecting the body systems, look at some substances that are under a black box by the TGA. That is, they still have their approval. They are still on that ARTG. They are still registered substances and accessible, but that there has been some published data about risks where the potential for that approval to be removed is very, very high. So all in all, the stages of drug development that we have been looking at, through pharmacognosy, high throughput screening, other methods, a candidate drug is discovered, or it might be an old discovery but a new lead for that substance. That would shorten the drug discovery time span, but if it was a new substance, it might be, say, up to 5 years while you find that lead. If it was that high throughput screening, you reject those chemicals that showed less potential for therapy or marketing, putting into a safe formulation, long shelf life, etc. You just optimize the good leads and start to do the pharmacology, profiling its risks and its benefits. A lot of that drug discovery might also overlap with preclinical data, where you then are really looking at those leads and those that might scale up to an industrial process. Even in complementary medicine, you might have to be looking at herbal preparations that could readily be used in a cream or a particular delivery system, whatever that formulation of the substance might be. And so, as with profiling, during the drug discovery stages where you're always interested in toxicity, now here in preclinical development you are trying to identify, either through animal studies or to save animals using computer simulations to look at the possibility of toxicology. And also we'll talk more about this term, pharmacokinetics, and during the preclinical trials starting to get a measure of what's happening within the body, but how the body is acting on that drug. Now, depending upon the substance, the researcher, their research funding profile, that might take add on a few years to that preclinical stage before you start the clinical development stages. Now, you'll look in your modalities as to which of these phases of clinical development of a pharmaceutical substance applies. We said here the phase 3, a large scale controlled clinical trial, might still apply to your modality, but that randomized controlled clinical trial, the golden standard, may not actually relate to your particular therapeutic substance. So phase 1 is just where you're looking at healthy volunteers. Here you are really testing that pharmacokinetics, how those healthy volunteers with their healthy liver, healthy kidney and other healthy body systems are handling that drug, that pharmacokinetics, how they tolerate it, already what adverse effects are starting to be seen. So that's a term that general practitioners might use with their clients, side effects, where that term can come under some criticism where the pharmaceutical companies are using it in preference to adverse effects, because side effects just makes it sound as if it's something minor and not really to worry about. Well, that's the perception that certain studies have shown that the general population has to the word side effects. The same effect on the body, nausea, described as an adverse effect, is always rated much higher than if you just called it a side effect. The very use of that term to the untrained layperson seems to suggest that it's minor and less worrying. Now, because some side effects are quite worrying, we won't use that term. We will always use the term adverse effects. And so it's from this phase 1 healthy volunteers that you start to get a profile of what those adverse effects might be. But it isn't until the phase 2 and phase 3 trials where you do get the full picture of effectiveness, that efficacy, and toxicity data. And now in phase 2 and phase 3 trials, where you're actually using it on patients that have that disorder, you're getting the true long-term toxicology profile. These people generally maybe not having such efficient heart, kidney or liver function. Okay, now once all of that data is available and published and available to the committees that report to the TGA, then we have that approval process where if the TGA or MedSafe and other similar regulatory authorities around the world approve that substance, it's then put onto a register in Australia, that ARTG, the Australian Register of Therapeutic Goods. But there's all sorts of review processes and those international protocols that Australia and New Zealand uses. Now, that process can take some time through the TGA, and then finally that substance available for marketing, where we said it's important for you to realise that it is still undergoing clinical studies where the general population is the patient now, where we're looking for, once again, still characterising rarer risks, the probability that a particular risk will show up in a population, unknown adverse events that could not have come out at any point through any of these testings may come out only when it's marketed to the general population. And so that information is still, as we said, on a regular basis fed back to that approval authority in Australia, the TGA, where approval can still then be removed for that therapeutic substance if the risks outweigh the benefits. But we just want you to get an appreciation for that timeline and add up all of those years, look for the worst case scenario, 5 plus another 1.5 years, plus 7 years, plus 2 years, so over 10 years of intensive research, paying researchers, and money then spent on marketing for that substance, where that pharmaceutical company wishes to recoup those costs through high sales of that substance. We've looked at different ways of classifying those substances once they are marketed and approved. That all of that trial data should be available to general practitioners, other allied health professionals such as yourselves in complementary medicine, and to the general public as well. The general public reports back to the TGA, and there are systems whereby they can do that either through their GP or independently now that we have electronic access to the Therapeutic Goods Administration. It's important to realise that there's another parallel with complementary medicine, that your clients can likewise report adverse events or adverse effects through to the TGA, or in New Zealand through to MedSafe. Here's this different classification system looking at those adverse effects. We'll use reactions to mean the same thing as effects. We'll call them ADR. Remember, your general practitioner's euphemistic term for that might be side effects. We'll also distinguish them from adverse events, ADE, and they are classified as follows. ADR type A are those that you could predict based on the pharmacodynamics of that therapeutic substance, which, as we'll see shortly, when we say dynamics, means how it actually acts in the body. So if there was an adverse event, you could have predicted that from the way that it works in the body. However, one of the reasons for doing population studies or even doing post-marketing surveillance is because some adverse effects on people are unpredictable, quite bizarre, and this would include allergic reactions and hypersensitivity reactions. Quite unpredictable, but you do have to do clinical studies to get a measure for the probability of that happening. And so that when marketing or prescribing this drug to a certain individual, they can then receive that information about the probability of a particular risk. Now, if it's a type C, that's a chronic reaction that could only have come out from those more long-term clinical trials on patients or from post-marketing surveillance where there's a chronic effect. It develops with long-term use of that therapy. And we'll study some of the drugs that show drug tolerance and a physical dependence, and we'll look at the pharmacodynamics of that in the body, these type C events. A lot of the drugs used to treat anxiety and so on, producing this drug tolerance. We'll also look at over-the-counter medications, things like aspirin and paracetamol, and how chronic long-term use produces these type C adverse drug reactions. With paracetamol, we'll also look at its toxicity profile, and in overdose how it is quite toxic, but we'll classify an overdose situation as being that adverse drug event, an ADE, to distinguish it from an ADR where it was taken in therapeutic and recommended amounts but still at those doses was shown to have a particular long-term effect. Now, some of these long-term effects might actually be quite delayed, so these type D events or type D effects might happen not necessarily to that generation that took the drug, it might be to their offspring. If it was the person themselves, it might be a delayed effect such as carcinogenicity. The drug or device was actually in the long term carcinogenic, caused cancer to develop or to progress. Then it might be teratogenic. It might be a delayed effect on the fetus at whatever particular trimester 1, 2 or 3, teratogenic. Might also come under the type D classification. Now, type E is end of use. Those adverse drug reactions that you see once you stop taking the medication, a withdrawal symptom. We mentioned drugs used to treat anxiety and how the person develops tolerance. You'll see that with these medications, another problem is when you stop taking them, the withdrawal symptoms that you see. So the drug tolerance was a type C classification, but the withdrawal symptoms were a type E, end of use. Now, one other last classification that's important, because it becomes important when we look at drug-drug interactions. Type F, adverse drug reaction, a failure of that therapy. One particular interaction that we're going to study in more detail later is a herb, St. John's wort, used to treat depression, and how it can interact with the oral contraceptive pill, leading to failure of the oral contraceptive pill. A type F adverse drug reaction. So these reactions or events reportable to the TGA, whether that substance, as we said, was a prescription medication, an over-the-counter medication, or a complementary medicine. Let's look in detail at some of those adverse drug reactions, the ADR types A to F. Carcinogenicity, as you'll consider, could come under several of those classifications, but we'll look at it as a delayed effect. And whereas we said that the substance itself may not have caused the cancer, that it may have simply led to the progression of that cancer. You'll study that particular mechanism of toxicity when you look at your first assignment. So these are substances whereby various physiological processes lead to either a cancer starting or to the progression of a cancer, And let's just have a look at a couple of interesting examples that other students have looked at when looking at potential toxins and looking for available research, looking at exposure and toxicology data on them. So certain air pollutants, asbestos, the actual early causative agent of a mesothelioma, and not only just that particular cancer, also implicated in some other effects, not necessarily cancerous, but an asbestosis, a disorder in the lungs, a fibrotic reaction, and so on. So you might have to study all of those mechanisms of toxicology, including the carcinogenicity aspects. Other people, because of their interest in nutritional medicine and healthy manufacturing of foods, are interested in nitrites, and they're an additive in food, and they would then look in detail at a whole variety of toxicology mechanisms, where one of the mechanisms is that it promotes cancers in the stomach and other places in the body. So important to distinguish when looking later at toxicology studies and then doing some in-depth research for yourselves, this idea of a mutagen. It may not necessarily be a chemical. And when you do your research on toxicology, which is the broader study, you'll look at a whole variety of other substances and agents other than chemicals that might be mutagenic. They actually initiate the whole process by damaging the DNA. So then something that is a carcinogen may not be the mutagen. It may not have actually directly started the process off and damaged the DNA, but a mutagen would be considered like a primary carcinogen. There may need to be a secondary carcinogen, something that is needed as well. The two combine to lead to the alteration of DNA or repair processes on mutations to the DNA. There might also be a co-carcinogen, some chemical that's needed to promote that process, and so a lot of the chemicals that you will research are just promoter substances, co-carcinogens, but still classified as causing that adverse drug reaction type D if it was a therapeutic substance that was the co-carcinogen. Now, similarly, all of these substances needed to promote and continue those alterations to the DNA. Then there are a whole variety of healing processes in the body, largely involving your immune system, that negatively stop, prohibit, block, shut down these processes and promote healing of the DNA and repair of the DNA and prevent progression to cancer. But then there may be actual promoter substances that overcome those body processes. And then it's more a cumulative thing, a cumulative exposure, maybe not to the primary carcinogen, but to some chemical in the diet or in a herbal remedy or some other exposure through the environment to that substance, where you then accumulate damage to certain key genes called proto-oncogenes, where once they become out of control and mutated and damaged, get converted into oncogenes. So those substances, promoters that promote the accumulation of damaged genes, these oncogenes, they might be called oncogenic substances, and we'll see that there are some viruses that are oncogenic. They may also be the primary mutagen that damages the DNA, but it would be that chronic exposure that then promotes the accumulation of these oncogenes to the point where the body's immune system cannot remove them from the body, and that you then get a tumour form, a transformation in those cells that is malignant. So in a similar manner, you will have to go into a little bit of in-depth research about other mechanisms of toxicity of substances. So if it is asbestos, that you'll look into how it causes the mesothelioma, what substance is it? Is it actually a primary carcinogen, secondary, or is it a co-carcinogen, or a promoter substance, and specifically stated for that particular agent? Or in asbestosis, it will then be a completely different mechanism of toxicity, which you will then investigate further in that research. Another important body process that you're going to need to talk about for a whole variety of agents will be apoptosis versus necrosis. This is an actual mechanism of toxicity that we want you to research to see if it might be relevant for your chosen agent that you're going to research. If it's apoptosis, to know that that's that programmed cell death where particular organelles in the body can then recycle components, particularly with DNA, the metabolism of the purine component of DNA, adenine and guanine, down to uric acid, and the recycling of those substances, returning them to the systems of the body, or perhaps excreting, in the case of the uric acid, excreted in the kidney, through the urine. However, if a substance leads to a necrosis, everything is lost. In apoptosis, the DNA was recycled. Here, everything. The DNA in the nucleus, every cell component, is suddenly released to the tissues or organs of the body. And a particular radical, nitrogen monoxide, also known as nitric oxide, which we're going to look at when we look at neurotransmitters. It's a particular radical in the body that's involved in mediating a lot of the necrotic processes. So when you study your particular agent that comes in from the environment, or might be in a herbal medication, or in our foods, the actual process that it damages the body might be via this nitrogen monoxide radical. So you'll research that in detail and report that it's a necrotic agent in the body. And last of all, relevant more to drugs, is this idea of a toxicity profile where substances are teratogenic. We've classified it as ADR type D, just a delayed effect. Here we have thalidomide as our example that we're using of a substance that shows teratogenic effects. This substance, thalidomide, was reported in the literature around about 1958 as having good sedative effects on pregnant women in the first trimester if they were experiencing a lot of nausea and vomiting. So the black line showing then the sales of thalidomide once these reports were published, and this substance was then marketed and available. The red line shows the studies done by McBride and others that reported a correlation. Prior to thalidomide sales increasing, there was certain levels of fetal abnormalities called phocomelia, so flipper-like limbs, arms and legs not developing properly, always a certain amount in the background. But the moment thalidomide increased in sales, the numbers of these babies being born with phocomelia suddenly on the rise, and a rise paralleling the thalidomide sales. So McBride and others then did the pharmacokinetics, further toxicology studies, and then showed not just that it was a positive correlation, but a cause effect, that the thalidomide was causing the phocomelia because it was actually teratogenic, damaging the buds that were developing in the fetus. So then population data continuing. This is pharmacosurveillance in action. Once thalidomide sales dropped off and eventually it was removed as an approved substance, the levels of phocomelia dropped away back to background levels. So today, thalidomide is available again and registered again, but for other indications. This time, certain rare skin conditions where it's immunomodulatory, certain cancers where, because of its immunosuppressive effects, thalidomide is indicated. But in all of these people, we have to establish that the person, if they're female, is not pregnant, because of this other pharmacosurveillance data available from the 60s, thalidomide remains category X in pregnancy, must not be used, directly teratogenic. And so some of these other effects, and why we're calling them delayed effects, we've used here as our example diethyl stilbestrol, and stilbestrol. Stilbestrol was shown quite clearly in population studies to have this type D delayed effect. If used during pregnancy by the pregnant woman, there was an increased risk of vaginal cancer, but not in that woman, in her female offspring. So now you can clearly see why it is a delayed effect there. And so you may be looking at environmental toxins that once again have a delayed effect on the next generation. So we also mentioned other ways of classifying therapeutic substances. Another way was the scheduling them, various levels of access depending upon their toxicity profile. So that's the keystone to this particular classification systems, that all substances, when used as medicines, are inherently toxic. It's just a matter of measuring that harm that they cause and coming up with a degree or the potential for that toxicity. It's a risk assessment. And that will determine what schedule and therefore what access, and will also then affect classification of that drug. So in Australia, scheduling is a state matter, but we have decided to come up with a uniform system, that there were some differences between states for the same substance. So now with this standard for the uniform scheduling of medicines and poisons, the current document that is used by the TGA and other health authorities, state and Commonwealth, we have ways then of controlling all poisons, including those that are therapeutic. So you'll see that this scheduling of poisons is an important consideration when it comes to medicines. They are themselves poisons and is relevant then to that TGA. Here's the schedules 1 to 9, and we'll just talk about a couple of them that will be important. When it comes to schedules 6, 7 and 8, these are going to be likely the substances that you'll be researching for your assignment on toxicology. Schedule 8 is a prescription substance, but generally has to be prescribed by practitioners where it has a potential for addiction and therefore abuse. We'll talk more about those substances such as pethidine and other opioid and narcotic substances later when we look at the drugs affecting the body systems. Schedule 1 is currently not in use. It was temporarily in use with Chinese medicines at one point. Now it's just not in use. It will be for those substances that in the doses sold are deemed to have such low toxicity. Schedule 2 is for a lot of over-the-counter medications, medications sold through the pharmacy, so it will include some supplements, some nutritional substances. Schedule 3 are those that don't need a prescription but can be sold. They'll still be over the counter, but they may require a pharmacist's advice, whereas Schedule 4 will require prescription from a general practitioner and other registered practitioners. Then last of all, of importance perhaps for your research and certainly for some of the substances that we're going to talk about as drug leads, prohibited substances, drugs that are prone to abuse. They may have that addictive potential with certain people, so therefore they have to be controlled, made illegal so that there is less access. But if you want to research one of them as a potential drug lead, that there's ways of applying for that research approval so that you can study Schedule 9 substances. So cannabis might come under that category.